So this is a multi-cohort, multi-center phase 2 study using a BCMA CAR-T cell product called Ciltacabtagene autoleucel, or Ciltacel. And this product was previously tested in other trials, the Legend-2 study initially in China, and then CARTITUDE-1, which established its efficacy in a US population of very heavily pretreated patients, triple-class refractory. And so based on that initial promising data, CARTITUDE-2 was started, and this is now looking at using the same BCMA CAR-T cell product, but in often less heavily pretreated patients and or in different populations of patients with unmet need. CARTITUDE-2 right now has 5 different cohorts, and what I was discussing at IMW was Cohort A, which was looking at using this BCMA CAR-T cell product in patients with 1 to 3 prior lines of therapy who were lenalidomide refractory. And so that's the basic background and setup of the study. It is designed to look at 20 patients per cohort, and the primary endpoint for each cohort is looking at the MRD negative rate, but obviously we're also looking for overall response rate, complete response rate, progression-free overall survival, and safety as well in these various populations. As mentioned, patients had to have 1 to 3 prior lines of therapy to get on the study, and had to be refractory to lenalidomide. They, once they enrolled, they had T cells collected by leukapheresis, and those cells were then manufactured to express the BCMA CAR. Patients could get bridging therapy during manufacturing, and this was at the investigator's discretion to try to maintain disease control during manufacturing. They then received fludarabine and cyclophosphamide as lymphodepleting conditioning, followed by a single infusion of the Ciltacel CAR-T cell product. And then they were monitored for safety and efficacy thereafter. Patients had the option of being treated as an inpatient or outpatient to get their CAR-T cells based on the institutional experience and preference. 20 patients were enrolled in the study, and this, as planned, was a less heavily treated population than CARTITUDE-1. So in this study, there was a median of 2 prior lines of therapy compared to a median of 6 prior lines for the prior CARTITUDE-1 study. About 2/3 of patients, though, were triple class exposed, meaning they had seen a previous proteasome inhibitor, IMiD, and CD38 antibody. 40% were triple class refractory, and all of them were lenalidomide refractory as required by the protocol. About 1/3 of patients had high-risk cytogenetics going into the study. And in terms of the looking at the primary efficacy results, the overall response rate was 95%, meaning 19 out of 20 patients responded. 1 patient had stable disease as best response. 75% of patients had a CR or better, and 85% achieved a VGPR or better. The median time to response was rapid at 1 month, and median time to CR was 1.9 months, so very similar to the CARTITUDE-1 study. Only 4 patients at this time were evaluable for MRD negativity. All 4 of them were MRD negative at the 10 to the -5th threshold by next-gen sequencing, but we're waiting further follow-up testing to assess the true MRD negative rate. And I think what was impressive so far, again, relatively short follow-up, only median follow-up of about 6 months, but no patient had progressed at the time of data cutoff, with the longest patient out about 1 year from CAR-T cells. So those are the primary efficacy findings. In terms of safety, overall, the safety profile seemed fairly similar to what we've come to expect with CAR-T cell therapy in myeloma patients. The primary toxicities included cytopenias, which were expected based on the lymphodepleting conditioning, although the frequency of high-grade, grade 3/4 thrombocytopenia and anemia seemed a little bit less in this cohort compared to CARTITUDE-1, perhaps because they were less heavily pretreated. Similarly, there seemed to be a lower incidence of prolonged cytopenias for more than 2 months. In terms of cytokine release syndrome, this was seen in 85% of patients, but all but 2 of these were grades 1 and grade 2. There was 1 grade 3 and 1 grade 4, and there were no treatment-related deaths. The median time to onset of CRS was 7 days, and that's one thing that differentiates this CAR product from some other ones where the median time to CRS is on the order of a day or 2. The ICANS, or typical CAR-related neurotoxicity, was seen in 3 patients, or 15%, all grade 1, grade 2, all resolved. after a median duration of 2 days. And I think importantly, there were no late neurotoxicity events in this cohort, particularly the Parkinsonian-like movement or neurocognitive treatment-related adverse events were not seen in this cohort of 20 patients. So I think this really just shows that certainly we can give these CAR T cells in less heavily pretreated population of patients. The efficacy seems similar to what's been seen with their use in later lines of therapy. And perhaps we're seeing a little bit less of the cytopenias, which was one of the hypotheses that when patients were not quite as beat up from prior therapies, they may recover a little bit faster. I think we're still obviously waiting for longer follow-up to see if the duration of responses is going to be longer when we treat less heavily pretreated patients compared to waiting until they've had a median of 6 or 7 prior lines of therapy, which has been done for most of the prior CAR T cell studies. So we need a little bit longer follow-up, but I think this really shows support for this idea of moving this up as an earlier line of therapy. And in fact, these data have been used to support the opening now of a randomized phase 3 trial for this same population, 1 to 3 prior lines, lenalidomide refractory, comparing Ciltacabtagene autoleucel or Ciltacabtagene versus doctor's choice of one of 2 standard of care therapies, either daratumumab pomalidomide dex or bortezomib pomalidomide dex. So that phase 3 trial will really serve perhaps to confirm these data that this is going to be a useful therapy for less heavily treated patients. So no, I already mentioned the phase 3 trial that is going forward as a follow-up to this cohort. This cohort is continuing to accrue, and there may be another 20 patients in the phase 2 CARTITUDE-2 program for Cohort A just to get a little bit more experience. And then obviously we're continuing to accrue to the other cohorts in CARTITUDE-2, which I think are exploring some interesting populations of patients, including patients who are progressing within 1 year of auto transplant or initial therapy. That's a poor prognosis group. There's a cohort for patients who have had a prior BCMA-directed therapy. So we'll get a sense of how useful this therapy is in that setting. And then there are 2 cohorts exploring the use of this. Really, it's consolidation for initial therapy, either for patients who have suboptimal response after an autologous transplant, or even instead of an autologous transplant, but doing this right after induction for high-risk patients. And so those cohorts are currently accruing, and hopefully we'll have some data over the next year or so exploring the use of cilta-cel in those settings as well.